Okay, great. We're going to get started on our next session, which is gene therapy advancements for CMT. This is a topic that is near and dear to my heart as both a human geneticist and someone who has spent 15 years working on gene therapies for sensory disorders. I'd like to start, though, by thanking Afrouz Rashananajad, who is unable to make it today, but invested a lot of time in curating this session for us. As you may already know, this work is quite cutting edge and holds a lot of promise for patients living with CMT, especially those with rare subtypes of CMT. Today, we're going to hear 4 talks, and we're going to start with Dr. Casey Maguire, Associate Professor of Neurology, Harvard Medical School, Investigator, Associate Professor of Neurology, Mass General Research Institute. Casey?
Thanks. All right. Can you hear me okay? Well, first, I'd like to thank CMTRF for the invitation to speak, especially Afrouz and Kate for putting all this together. It's a real pleasure to be here with you today. Let's see if I get this to work. So I'm going to talk about some recent developments in our lab regarding gene delivery to peripheral nerves. Here's my disclosure slide. So I hope I can at least convince folks in the room that different routes of administration for gene delivery systems, including adeno-associated virus, are promising. Although they both have advantages and disadvantages, yet we can improve upon those, mitigate some of those disadvantages, improve the pros using technology development. So for intrathecal delivery, that's a popular method of AAV delivery, especially for the spinal cord. The pros and cons are, I kind of made this little happy and sad chart here. It's very subjective, but you can see that this is working. The virus has to, or whatever you deliver into the intrathecally, has to travel long distances to reach the distal nerves, which are affected in CMT. So the distal nerve biodistribution is certainly a limitation. I mean, it does still work. It's just a matter of how efficient it is. Antibody levels, while not completely avoidable with antibody levels, I mean antibody levels to AAV, to the virus that we work with. It's less of a concern because you're not delivering into the peripheral circulation. And then again, because you're giving more of a local CSF compartment delivery, the peripheral toxicity is less. Although I'll tell you that there's plenty of studies done with intrathecal AAV and 90% of the vector still enters the periphery. So it's not exactly completely not an issue. We've done a small study in non-human primates a few years ago. This is unpublished, where we injected a pool of AAV vectors intrathecally, and then we looked at the vector genomes. That's what the virus is actually delivering for gene therapies, right? We looked at in different nerves, some close to the injection site, like the cranial nerve and the sciatic nerve, and also in more distal nerves, like the ulnar nerve and sural nerve. Those are on the further. Oh, sorry about that. Figure out what I'm doing here. All right. So essentially, you can see in both animals, there's different grades in different animals, which is common in non-human primates, but there's a big gradient. There's a 13-fold drop off and a 5-fold drop off in the second primate. So with systemic delivery, there's also pros and cons. First of all, when you're delivering it IV, you have better biodistribution throughout the body and potentially a better access to distal nerves. However, you have much more susceptibility to the immune system and anti-AAV antibodies. And again, as many people have heard in the news and headlines, high dose AAV is associated with systemic toxicities in some cases. So that's certainly a challenge. Although again, we can improve that likely by increasing the efficiency of the vector, lowering the dose. So first, I'm going to talk about a capsid that we engineered that we found can deliver to peripheral nerves in mice so far. So we did an in vivo selection. This is kind of a standard AAV9 peptide library selection in mice. We were doing a neurofibromatosis type 1 model. That's what we were interested in at the time. But we took both the naive nerve and the tumor-bearing nerve and did 2 rounds of selection. So we basically did survival of the fittest, found the best capsids in the mouse system after IV delivery. And what we found here, I've highlighted a couple. We did this is the second round NGS data, some of the top capsids from that data. And we did 2 animals during that second round, one on the tumor side and the naive side for both animals. And you can see in the green and the magenta, there's some common peptides in either both tumors from both mice or on the naive and the tumor from a given mouse. So we took those top hits, so the top candidate capsids, they're called SC1 through 4, and then we compared them to the parental virus. That's kind of the benchmark capsid for that experiment. And we put them through wild type mice. So at this stage of the game, we weren't really we weren't working in the NF1 model. We're looking at kind of like healthy, naive nerve. And what was pleasantly discovered was that one of these capsids, AVSC4, that's showing up over here, was in terms of frequency of these barcodes of these genomes, was going to the sciatic nerve much more efficiently, about 6-fold better than the standard AV9, which does traffic to the nerves after IV delivery. So it's not like AV9 is completely not able to do that. So then we did a side-by-side, like head-to-head validation study in, again, naive mice, 6 mice, male, female, injected IV. We used a P0 promoter in this experiment, and we looked at genomes and also mRNA to look at expression. And on the top, you can see that we backed up our initial data. We saw a 6-fold increase over AV9 with SC4. We were really disappointed when we saw the transduction data, which really showed no difference between the capsids. So we were scratching our heads. We decided, like, look, it might be a promoter issue. That promoter's active in certain subtypes of Schwann cells. So we went ahead and we did another experiment where we used a CAG promoter, which is much more broadly active than the P0 promoter, and we injected mice IV. And what we found was we could transduce the nerve, and then we did SOX10 staining, and we found colocalization with SOX10 positive Schwann cells. So this capsid does indeed transduce Schwann cells after IV delivery. So then after doing that, we actually looked at what that peptide was that was displayed on the capsid. It's this GATP STQ motif. And when we started doing a literature search on that, the STQ motif is a well-known laminin binding peptide that's been identified through phage display by actually a couple independent groups over the years. Actually, one group found that it binds to denatured laminin more than native laminin. So, but in any case, we'll come back to that. So, what is laminin? I had to kind of go back to the cell biology books to remind myself. We think it's a promising target for AAV delivery to the nerves and maybe other modalities. It's highly expressed in the basement membrane of Schwann cells and peripheral nerves. There are like 15 types of laminin because there's different. They're basically a trimer of it gives this cruciform structure here, as you can see right here, and the image is a little blurry, but that's from a publication that shows laminin staining around the nerve in red. So laminin's in green. You can see it completely like encircles the nerve. So there's just a lot of laminin on peripheral nerves. This other group actually showed that targeting, they found, again, a peptide that had a similar motif, NTQ, and that could actually exquisitely target peripheral nerves in mice in another study. So one thing is we were like, well, laminin's expressed everywhere, right? So it's probably not, the capsid is probably not going to be very specific at all. That was kind of our initial guess. Through a recent collaboration, we did a comparison of AAV9 with SC4 after IV delivery. And to our surprise, at least in this preliminary study, which needs to be repeated, we found lower levels of transduction in several tissues, including muscle, which has a lot of laminin expressed. So that was promising in terms of having a capsid that would hopefully be more targeted to your tissue of interest. We looked recently at we found that the peptide does indeed bind laminin using what's called dot blots, where we spot laminin and we added biotinylated peptides. The biotin SC4 is our peptide. It bound to human and human laminin 1 and 2, and the positive control also bound, and a scrambled peptide did not bind. Interestingly, found that we did not find binding to mouse laminin, which doesn't make a lot of sense. So we're looking into if there's an issue with that protein or whatnot. But it was encouraging that it binds to human laminin, suggesting its translational potential. Again, this is just another binding curve that a scientist in my lab did to show strong binding to human laminin without binding to mouse laminin, at least in this assay. Although we're going to try to denature it to see if that it's something with a cryptic epitope or something like that. How am I doing on time? That's
a really great question.
Okay. I don't see a time.
Minutes.
5 more minutes. 5. Okay, great. So to conclude from this section, directed evolution identifies an AVSC4 capsid displaying a laminin binding motif. It transduces Schwann cells after IV delivery. We saw lower off target in a pilot experiment. It binds to human laminin, and then we'd like to do some translational studies. So you can see that from before, we've improved the biodistribution. Hopefully, the peripheral toxicity, that'll have to be determined down the line. Of course, antibody levels, that we didn't address that issue in this. So the second part of the talk, we're going to talk about the other route of administration that's popular to get to peripheral nerve, which is intrathecal delivery. So this is a publication from a year or so ago that we did where we did another selection, but we did it directly in non-human primates. And then we isolated. We were interested in that collaboration and getting capsids that would biodistribute better in the spinal cord. And we did find that. So this is, you can see AAV9 is on the far left, and then these other capsids are kind of our novel 7-mer capsids. We could see up to 100-fold better biodistribution in the spinal cord. And what was really interesting, we saw like hundreds to 1,000-fold less biodistribution in the liver. As I mentioned, you say, oh, it's an intrathecal delivery. Well, actually, there's a ton of vector that ends up in the periphery. So we do think that's a promising part of these capsids. Hopefully this will work. And we actually showed that we got good transduction in human spinal cord organoids using some of these top candidates as well. So it wasn't a primary goal of this project, but we were interested in looking at some of these capsids in the peripheral nerves and the distal nerves. So we looked at, this is a kind of a heat map of the different capsids, which are shown here. AAV9 is kind of your benchmark, right? And then there's different, you probably can't see it, I apologize. The distal nerves are over on the right, and the more, the cranial nerve and the sciatic nerve are kind of on the far left. So what we found was that this one capsid, DR2, displayed a 4-fold higher frequency in trafficking biodistribution to the ulnar nerves when we normalized the frequency from the barcode data by NGS. So to conclude, directed evolution in NHPs identifies capsids with enhanced biodistribution to distal nerves, and we hope that we'll have lowered off-target biodistribution to liver using these capsids as well. So again, before we've improved the distal nerve biodistribution, antibody levels, again, wasn't a problem. And hopefully we've reduced some of that liver peripheral transduction that's been seen. Actually, another group at Novartis showed that it's still intrathecal delivery could cause liver damage to some extent in non-human primates. So to conclude, every route of administration has advantages and disadvantages. However, tech development can lessen the disadvantages. SC4 mediates higher biodistribution to peripheral nerves via the STQ motif and combine human laminin, making it potentially translation relevant for human gene therapy. And selection of the capsid library in non-human primates via IT delivery identifies capsids that can travel over long distances to distal nerves. So with that, I'd like to acknowledge all the people in my lab now and over the years that really have contributed and done a lot of this research, including former lab members And also, I should mention the former Swan Bio team, who is a really great collaborative group of scientists that worked on that NHP intrathecal selection study. And finally, the Gilbert Family Foundation, who supported the work that led to the identification of the SC4 capsid. So with that, thanks a lot. I'm not sure if we're doing questions now or later.
Yeah, we're going to do questions now.
Okay,
We can take
great.
5 minutes of questions.
All right. Well, thank you very much.
So I hate to ask a really naive question that shows I don't know anything about this, but so intrathecal means you're going in basically in the spinal cord. Is that?
Yeah, into the like lumbar intrathecal space.
So
Yeah.
does that mean you've already reached the blood-nerve barrier at that point?
I think so. I believe so. Yes. Yep.
Okay, so
Yep.
even though that's considered central nervous system, there's a pathway into the peripheral nerve.
Yeah, that's a great question. Like, I don't think folks really understand how AAV traffics along nerves. We know that AAV can actually go up or down an axon. People have shown that in different experimental settings. But it could also traffic through CSF. It could go to the periphery and then come back across the blood side. It's not really, to be honest, it's not well understood. Yeah.
Here, over here. So maybe another naive question, so forgive me if it's really naive, but you said it's traveling the AAV, but is it actually traveling, or is it just that you have a better binding to is it active transport, or is it actually just the peptide allows you to actually, if it travels there by whatever mechanism, it binds there?
Yeah, that's a good question.
And it relates to another question I have is,
Yeah.
and again, I'm not working in the gene therapy field,
No
but
problem.
you're using mouse, right? A few millimeters, and you show your human skeleton, and that's meters, okay?
Right.
And I did once learn from a neurologist that when you look for CSF levels down here, or you go up here,
Yeah.
there's actually a gradient. And I was told it's even whether you move or lying flat.
Yeah. Yeah.
So the point I'm trying to ask is, what can you translate from these small animals when you then go to a large animal, us?
Right.
And how because of the transport. So is there a way that you actually have active transport, or is it just gravity?
Right. So that was a lot, but I'll go for the second one first. So, all right. First of all, the intrathecal studies were done in monkeys. Yeah. But a monkey is still an old world monkey that's a cyno is still like, it's like a cat. Yeah. So it still has to travel much longer distances than humans, but it models it better than a mouse does. So it's still, I think you have to test it in the best models you have, of course, but it's going to have to, I think the monkey is way better than the mouse model because it's very small distances in the mouse. Regarding your question about like gravity and everything else affecting biodistribution throughout the CSF and the spinal cord, I think that has been shown in different biomolecules, including AAV, to make a difference. I think in some intrathecal AAV trials, they actually use the Trendelenburg positioning, where they basically put patients kind of like upside down to get AAV to move by gravity better. So that can affect it. But the virus can also, it's been shown, it can traffic on neurons. To your first question about do we have evidence that shows it's better biodistribution by actually trafficking there, or is it just binding there and like you have more AAV that stays around, maybe it doesn't get degraded, the genomes? Yeah, no, we don't have the exact mechanism down. So that's a good question. Yeah. Julia.
Hello? Hi.
Yep.
Sorry.
Oh, yes.
I can go next. Yeah.
You can go ahead.
Okay. So just to follow up the biodistribution question.
Yeah.
In your library of capsid with intrathecal approach, have you looked at the cell-specific tropism?
No.
Neuron versus Schwann?
No.
Yeah.
No, unfortunately, we really couldn't in that study. It was really a pilot study to lead up to candidate selection to really know how those vectors perform. It was like 14 capsids pooled together. We're going to have to do with some funding, hopefully, a caps head-to-head capsid study with individual capsids per animal, looking at like reporter gene expression, what cell types. So we don't really know from that. Sorry.
Hi, Casey.
Hey, how you doing?
So you've designed some beautiful AAVs throughout your career, including some that we've used in the inner ear.
Oh, that's great.
We don't even have to worry about delivery. It's right there. And you can see that it's cell-specific
Yeah.
tropism.
That was a little
So
easier.
there's a lot of clues there, like in getting hair cells versus supporting cells. But I'm a little unsure about what the distribution in different cell types when you do an intrathecal injection or a systemic injection. I can understand the practicality of wanting to only express something in certain cell types, but I'm much less clear on whether this has any bearing on immune response. So I want to hear your thoughts on how this affects immune response and concerns about repeated dosing. And then I would like your thoughts on what about mutations where maybe the main pathology is a peripheral nerve, but in reality, every cell is in one way or another suffering potentially from these mutations.
All right, another good question, set of questions. Hopefully I have a little bit of time, Yuri, to answer your question, but I'd say, yeah, no. I think if you're saying what's known about like the preferential route to avoid immune reactions, is that kind of what you're getting at?
I think it's kind of related to the earlier question about, so we can see that specific tissues are expressing the virus.
Sure.
But does that mean that the virus is somehow only accumulating there? Or is it all over the place and it just happens to get absorbed by the cells in those specific regions? How much do we know about how much the virus is traveling throughout the body, even if it's not getting into the cells?
Sure. That would have to be done with other, like, again, for like like that capsid right now, we'd have to do like, let's just focus on the SC4 capsid for IV delivery. We haven't done very careful biodistribution studies looking at genomes throughout the body. But we have looked at the liver and brain, and I mean, it is going everywhere for sure. So I think with AAV, people throw around hopefully did I say targeting in this talk? Probably I did, but I think you throw around that word a little loosely sometimes. We as a field throw around, it's gradients, right? It's not like 100% targeted to the nerve for sure, but it's much more efficient than AAV9, at least sixfold. I think that's a good start, but certainly there's a lot of vector that goes body wide. So it's still a concern. Like, what is the threshold going to be before you have these systemic toxicities? I mean, there's plenty of times where people have used IV administered AAV and there haven't got any toxicity. So in general, it's like 1E13 VG per kg or lower in general. But I think it's going to depend on a lot of things like the serotype, the patient, and probably your transgene you're expressing too. But probably we don't have time to go through everything today. But I'd love to talk to you after, Yuri. Yeah, especially about the inner ear. All right. Well, thank you very much. Yep.